Hello friends, in the last lecture we have completed chapter amines, and today we are going to start our new next topic, that is biomolecules, in which we are going to start with carbohydrate. First we have to understand what are biomolecules. So biomolecules, every living organism like plants or animals requires large number of many molecules which constitute their structure and to control various physiological processes. So for human being, many molecules required for our development of bone, development of muscle, then working of many glands like pancreas secrete insulin or hormones, etc. All these molecules, they are called biomolecules. They are lifeless molecules, but they require for our life. They are very essential for every living organism, that is for plants and animals. Such molecules are called biomolecules. There are three important biomolecules we are going to study in this chapter. That is, first carbohydrates, second is protein, and third is nucleic acids, that is fats, oils, lipids, etc. So all these molecules, they are very essential for every living organism. Therefore these molecules are called biomolecules. So let us start with our first very essential molecule, that is carbohydrates. So for what are carbohydrates? Carbohydrates are polyhydroxyaldehydes or ketones. That means they contain aldehyde group, that is CHO group, or ketone group, that is C double bond O group. And these molecule contains polyhydroxy group, that means more than 2 or 3 OH groups. The polyhydroxyaldehydes or ketones are called carbohydrates, or the compound which gives such units on hydrolysis are also called carbohydrate. For example, sucrose or cane sugar on hydrolysis gives glucose. Therefore cane sugar or sucrose is also called carbohydrate. Or starch, which is on hydrolysis gives glucose, therefore starch is also called carbohydrate. So any molecule which on hydrolysis gives polyhydroxyaldehyde or polyhydroxyketone are called carbohydrate, and polyhydroxyaldehyde or ketone, they are also called carbohydrates. Carbohydrates are also called saccharides, and therefore in the classification you can see they are classified as saccharides. So the second name of carbohydrates is saccharide. Example of carbohydrates are glucose, fructose, starch, or maltose, or lactose, galactose. Many carbohydrates. There are more than 200 carbohydrates are known up till now. We are mainly going to study about glucose. Glucose has a chemical formula C6H12O6. So carbohydrates are the polyhydroxyaldehydes or ketone, and the compound which gives such unit, or the compound which gives polyhydroxyaldehydes or ketones on hydrolysis, are also called carbohydrates. Now let us study the classification of carbohydrates. Carbohydrates are classified as, mainly classified into three groups: monosaccharides, oligosaccharides, and polysaccharides. Mono means one, it has a one basic unit, or on hydrolysis gives, which cannot be hydrolyzed. Oligosaccharide are the compound which on hydrolysis gives 2 to 10 monosaccharide units, and the polysaccharides are the carbohydrate which on hydrolysis gives more than 10 monosaccharide units. But monosaccharide unit cannot be hydrolyzed further. So the detailed classification is shown in this tree diagram. Carbohydrates or saccharides are classified into, first is monosaccharide which do not hydrolyze into smaller unit. Example, glucose, fructose, ribose, etc. Oligosaccharides are the compound which yield or which gives 2 to 10 monosaccharide units on hydrolysis. And polysaccharides are the compound which gives many monosaccharide units or large number of monosaccharide units on hydrolysis, or you can say more than 10 number of monosaccharide units. Example is starch, glycogen, cellulose, etc. Now, depending upon the number of monosaccharide units, oligosaccharides are further classified into three groups. First is disaccharide, which gives 2 monosaccharide units on hydrolysis, or trisaccharide, which gives 3 monosaccharide units, tetrasaccharides, which gives 4 monosaccharide units. Similarly, if question is asked what are pentasaccharides, that means the carbohydrate which gives or which yields 5 monosaccharide units are called pentasaccharides. So question may be asked in the exam, define disaccharides. Disaccharides definition is the carbohydrate which yields 2 monosaccharide units on hydrolysis are called disaccharide. Example is sucrose, which on hydrolysis gives 1 glucose and 1 fructose unit. Second example is maltose, which on hydrolysis gives 2 glucose units, or lactose, which on hydrolysis gives 1 glucose and 1 galactose unit. Similarly, if the question is asked what is trisaccharide, what are trisaccharides? Definition is the carbohydrate which yields 3 monosaccharide units on hydrolysis are called trisaccharides. Example is raffinose, which gives 1 unit of glucose, 1 unit of fructose, and 1 unit of galactose. Similarly, tetrasaccharide example is stachyose, which on hydrolysis gives 1 glucose, 1 fructose, and 2 galactose unit. So this is the classification of carbohydrate which is already given in your textbook. Now we are mainly concerned about glucose. So the next study will be more of the glucose. Now, how these carbohydrates are nomenclature of carbohydrates. This glucose, fructose, raffinose, galactose are the common names. Now, first structure you can see, this is the structure of glucose, in which on the top there is a CHO group, aldehyde group, and below aldehyde group there is 4 CHOH groups, and finally there is a CH2H. Common name of this compound is glucose, but the IUPAC nomenclature of this compound is aldohexose. Aldo means it contains aldehydic functional group, and hexose means it contains 6 carbon atoms. You can count, there are 6 carbon, 12 hydrogen and 6 oxygen. The molecular formula is C6H12O6. I have shown here the structural formula. So glucose is also called aldohexose. Similarly, the second structure is fructose. You can see there is a C double bond O group, that is, there is a ketonic group, and if you count the molecular formula, there are 6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms. So glucose and fructose are isomers of each other. They have same molecular formula, that is H12O6. But in aldehyde, in the glucose, the functional group is aldehyde, but in the fructose, the functional group is ketone. Therefore, fructose is a common name, but IUPAC name is ketohexose. Keto means ketonic group, and as there are 6 carbon atoms, it is called hexose. Similarly, third example, it contains 5 carbon atoms, an aldehyde group. Therefore, IUPAC name of this compound is aldopentose because it contains 5 carbon atoms. These are few examples for your homework. You have give IUPAC names of the following monosaccharides. First example is, I will just give hint, it contains 3 carbon atom and aldehyde group. Therefore, the name of the compound is aldotriose. Second example is aldopentose. Third example, you have to identify the name of this third example. Only hint is it contains ketonic group, so the name will start with keto. So use your brain power and give names of these 3 structures. Now let us study the important part, that is preparation of glucose. Many times question has been asked, explain the laboratory method of the preparation of glucose. So in the laboratory, glucose is prepared from sucrose or cane sugar. Cane sugar or sucrose has a formula C12H22O11. When cane sugar is hydrolyzed with water, reacted with water, in the presence of H+, it may be hydrochloric acid or sulfuric acid. So when cane sugar or sucrose is heated with water, that is hydrolyzed in the presence of HCl or H2SO4, a mixture of glucose and fructose is formed. C6H12O6 plus C6H12O6, but glucose and fructose mixture is formed. So to separate glucose from fructose, during the cooling of this mixture, ethyl alcohol is added. Therefore, on the arrow C2H5OH is written. So what ethyl alcohol does, glucose is completely insoluble in ethyl alcohol C2H5OH, hence it crystallizes out. Only the crystal of glucose are formed, but fructose is completely soluble in ethyl alcohol, hence fructose remains in the solution. Therefore, when this solution is filtered, only crystals of glucose get separated from fructose. So question might be asked, why ethyl alcohol is used in the laboratory method of preparation of glucose? The answer is, in this reaction a mixture of glucose and fructose is formed, and when ethyl alcohol is added, fructose remains soluble in ethyl alcohol and therefore it remains in solution, and glucose is insoluble in ethyl alcohol, therefore the crystal of glucose are formed and they get separated by filtration. Hence ethyl alcohol is used. Now second method of preparation of glucose is from starch. This is industrial method or commercial method of preparation of glucose. When starch, that is C6H10O5 n times, many these C6H10O5 units are linked with each other. When starch is heated with water in the presence of sulfuric acid at 393 Kelvin at 2 to 3 atmospheric pressure, the condition is 393 Kelvin, very important to remember, and the pressure is 2 to 3 atmospheric pressure. So n number of glucose molecules are formed. So you can see in this reaction no fructose is formed, therefore no need to separation. Therefore large number of glucose molecules are formed. Therefore this method is used as a commercial method of preparation of glucose. In the next lecture we will see the chemical reaction and physical structure of glucose.